Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's fascinating topic, please take a moment to like, subscribe, and share this deep dive with your friends and family. Your support really sustains the Science Experience. Okay, so today we're taking a deep dive into something pretty fundamental: the molecular basis of aging, disease, and specifically the role of mitochondria, the cell's power plants. We've got some research here that seems to, well, challenge a really long-standing idea about how mitochondrial failure actually causes problems.
That's right. It's a big one. I mean, for decades, the standard view, the one in the textbooks, has linked mitochondrial dysfunction directly to aging and all sorts of complex diseases. The idea is usually that mutations build up in the mitochondrial DNA, the mtDNA.
Right, and that mtDNA holds the instructions for some really crucial proteins, doesn't it?
Exactly. 13 essential protein subunits for the oxidative phosphorylation system. We usually call it OXPHOS.
That's the system that makes most of the cell's energy, the ATP.
Precisely. So the classic story goes like this. mtDNA mutations mess up those 13 proteins, leading to defects in the RxPHOS complexes, like complex 1, complex 4, and so on. And the result? A quantitative loss of energy, a bioenergetic defect. The cell just runs out of juice.
Like a factory slowing down because the power keeps flickering? Makes intuitive sense.
It does. But this new work asks a really critical question. Is that decline in the cell's health and fitness really just because of the power shortage, the lack of OXPHOS capacity, or is it maybe due to something else? Specifically, the errors themselves, the mistakes generated when the cell tries and fails to synthesize proteins from those faulty mtDNA instructions.
So the process of making the broken parts might be more damaging than simply lacking the working parts.
That's the core question. Is the toxic waste worse than the lack of output?
Okay, that's a fascinating angle. So how did they possibly separate those two things, the energy loss versus the toxic translation errors?
Well, they needed a clever experimental setup. They started with a well-known model system, mouse embryonic fibroblasts, MEFs, from the Polg D257A mutator mouse.
Ah, the Polg mouse. That's the one with the faulty mitochondrial DNA polymerase, right? The proofreading part is broken.
Exactly. So its mtDNA polymerase makes tons of errors, leading to a rapid random accumulation of mtDNA mutations. It's a classic model for studying accelerated aging phenotypes driven by mitochondrial problems.
So you've got cells that are basically guaranteed to have corrupted mitochondrial blueprints. Step one, check. Now, how do you isolate the effect of trying to read those bad blueprints?
That's where the specific tool comes in, an antibiotic called chloramphenicol, or CA.
Chloramphenicol, isn't that used clinically sometime?
It has been, but its key property here is that it specifically inhibits mitochondrial translation. It stops the mitochondrial ribosomes from making those 13 proteins encoded by mtDNA.
Okay, so it hits just the mitochondrial protein factory, not the main protein factory in the cytoplasm.
Correct. It acts as a very specific brake on mitochondrial protein synthesis. So treating cells long-term with CA effectively shuts down the assembly and function of the OXPHOS complexes. It creates a severe energy challenge, mimicking the outcome of mutations.
Wait, if you block OXPHOS completely, wouldn't the cells just die? They need that energy pathway?
Usually, yes.
Okay.
And that's the crucial control. To keep the cells alive despite the CA-induced energy block, they supplemented the growth medium with uridine.
Uridine, a nucleoside, how does that help?
It allows the cells to bypass a specific metabolic requirement linked to the respiratory chain, the synthesis of pyrimidines. With extra uridine, the cells can survive and grow, even with severely impaired OXPHOS function.
Got it. So uridine is like putting the cell on life support, metabolically speaking. It keeps the lights on even though the main power plant is offline because of CA?
Precisely. And this setup allows for the key comparison. You take the Polg mutant cells, which are already struggling with bad mtDNA instructions,
and you treat them with CA plus uridine. So now they have both the underlying mutations and a complete block on translating any mitochondrial proteins, faulty or not.
Exactly. So the logic is, if the cell's decline was primarily due to the lack of energy from faulty OXPHOS, then adding CA, which also blocks OXPHOS, should make things worse, or at least no better.
But if the decline was primarily due to the stress and toxicity of trying to translate the faulty instructions,
then blocking that translation process with CA, even though it maintains the energy deficit, might actually improve the cell's molecular health.
Okay, so stage is set. So what happened when they actually ran this? Did the Polg cells decline as expected without CA?
Oh, yes. They cultured the Polg D257A MES long-term and saw exactly what you'd expect. mtDNA mutations piled up, mitochondrial protein synthesis rates dropped, and crucially, they observed detrimental changes in nuclear gene expression and the overall cellular proteome.
Or specific markers going the wrong way?
Yeah, things like the stem cell marker CD90 changing, and changes in STAT1, which is involved in innate immunity signaling. Clear signs of declining cellular fitness and stress.
Okay, baseline established. Now for the CA paradox, as you called it. They added chloramphenicol plus uridine.
And the results were, frankly, quite remarkable. The CA treatment, blocking mitochondrial protein synthesis, significantly reversed many of those molecular signatures of decline.
Wait, reversed? You mean things got better at the molecular level?
Yes. The accumulation of mtDNA mutations continued. Of course, CA doesn't fix the underlying mutation problem. But the expression of those mutations at the RNA level and their subsequent translation into proteins, blocking that process seemed to alleviate the downstream pathology.
So shutting down the factory producing faulty goods improved the cell's overall molecular state, even though it meant the factory produced nothing and the energy crisis persisted.
That's exactly what the data suggests. It strongly implies that the process of translating corrupted mitochondrial messages is, in itself, a major source of cellular stress and dysfunction, perhaps even more so than the resulting energy deficit alone.
That really flips the script. It's not just the lack of the final product, it's the toxic process of trying to make it from bad instructions.
Precisely. The errors generated during synthesis seem fundamentally different and possibly worse than simply inhibiting synthesis altogether.
Wow. Okay, so if this translation error is the real villain, does that explain other things we see in these aging models? Like changes in metabolism or that chronic inflammation?
It seems to tie in directly. Let's look at metabolism first. The reversal of those negative molecular signs by CA didn't happen in a vacuum. It required a distinct metabolic shift.
The cells had to compensate for the energy block somehow, right? Especially with the uridine support.
Right. And what they found was that the CA-treated Polg cells showed a really robust increase in the levels of a specific metabolite called Alpha-ketoglutarate, or AKG.
AKG. That's a really interesting one. It's not just metabolic fuel, it's heavily involved in epigenetics, right? And stem cell function.
Exactly. It's a key player in regulating enzymes that modify DNA and histones, influencing gene expression. So the cell, faced with this severe OX-PHOS block imposed by CA, seems to rewire its metabolism to boost AKG.
Which might help stabilize the cell, maintain its identity, or manage stress epigenetically, even while energy production is crippled.
That seems to be the implication. It's a strategic metabolic adaptation to cope with the translation shutdown, diverting resources towards stability rather than futile protein synthesis.
Okay, that's metabolism. What about the immune side? You mentioned STAT1 earlier, and chronic inflammation is such a hallmark of aging.
Yes, and mitochondria are critical signaling hubs for the innate immune system. One of the major problems in the Polg mutator mice, and presumably their cells, is this persistent activation of type 1 interferon signaling. It's like a constant state of antiviral alert.
Even without a virus, how does that happen?
It's thought to be triggered by mtDNA instability. When mitochondria are damaged or stressed, bits of mtDNA can leak out into the cytoplasm. The cell detects this misplaced DNA using pathways like CGA sting, which normally detect viral DNA.
So the cell essentially triggers an autoimmune or autoinflammatory response against its own damaged mitochondrial components.
That's the idea. It leads to chronic low-grade inflammation. But here's the kicker. CA treatment in these Polg cells robustly reversed these type 1 interferon signaling phenotypes.
It calmed down the false alarm.
It did. And it also restored the function of NRF2, a key regulator that acts like a rheostat balancing inflammation and oxidative stress response.
That's pretty strong molecular evidence. Did they test the functional immune response?
Yes, they did a classic challenge. They exposed the MEFs to bacterial lipopolysaccharide, LPS, which normally triggers a strong inflammatory response.
And the untreated Polg cells presumably overreacted.
Massively. They secreted excessive amounts of the pro-inflammatory cytokine interleukin-6, IL-6, a real hyperinflammatory response.
But the CA-treated ones?
The CA treatment brought that IL-6 secretion right back down to the levels seen in normal wild-type cells. It essentially normalized the inflammatory response.
So silencing the toxic translation didn't just fix internal molecular markers, it actually restored a more balanced immune function.
It appears so. By stopping the production of whatever aberrant signals were driving that chronic interferon state, CA allowed the immune signaling pathways to reset.
This really paints a picture where the translation process itself is a major source of stress signals. Did they connect this directly to sort of general cellular stress pathways?
They did. They linked it to the integrated stress response, or ISR. This is a major pathway cells activate under various kinds of stress, often leading to a general shutdown of protein synthesis and activation of survival programs.
And they think faulty mitochondrial translation triggers the ISR?
Yes. They showed that messing with mitochondrial protein quality control specifically activates the ISR. For instance, inhibiting a protease called AFG3L2, which helps degrade faulty nascent proteins inside the mitochondria, was enough to switch on the ISR.
So the cell detects the buildup of bad proteins or the struggle to make them and hits the alarm bell.
Exactly. And they use another tool, a molecule called actininin, which causes mitochondrial ribosomes to stall. Basically, it jams the assembly line.
Inducing a translation defect directly.
And doing that caused the cells to pump out huge amounts of GDF-15.
GDF-15, that's increasingly recognized as a biomarker for mitochondrial stress and dysfunction, often elevated in aging and various diseases.
It is, but here's the crucial link. If they treated the cells with actinin and chloramphenicol simultaneously.
So stall the ribosomes with actinin, but also completely block any translation with CA.
The GDF-15 secretion was completely blocked.
Wow. So the stress signal, GDF-15, wasn't triggered just by the ribosome stalling. It required the attempt at translation to still be happening, however poorly.
It strongly suggests that the ISR activation and GDF-15 secretion are direct consequences of defects in the mitochondrial protein synthesis process itself, independent of the downstream energy consequences. Stopping the faulty process stops the stress signal.
Okay, let's try to pull this together. This research seems to propose some pretty fundamental shifts in how we should think about mitochondrial disease and aging.
I think it points to at least 4 key principles that challenge that classic bioenergetic view.
Okay, lay them out for us.
First, the simple loss of OXPHOS energy capacity, while clearly not good, likely doesn't explain the full picture of pathology we see when mitochondria fail.
Right.
There's more going on.
Right. Second,
the errors that happen during the translation of those mitochondrial proteins seem to be fundamentally different, and potentially much more toxic than just the absence of the final product caused by complete inhibition.
so making mistakes is worse than doing nothing in this context.
It appears so. Third, these translation defects act as potent stress signals. They trigger chronic inflammation, like the type 1 interferon response, and lead to the secretion of cytokines like IL-6 and stress markers like GDF-15, affecting cell communication.
Spreading the stress signals around. And fourth?
Fourth, paradoxically, completely inhibiting OXPHOS, as they did with CA, forces this compensatory metabolic rewiring, like boosting AKG, which might actually help the cell cope by influencing the epigenome and managing stress, even under severe energy limitation.
So the core takeaway message is really about the fidelity of mitochondrial protein synthesis. Quality control is paramount, and errors in that process are a major driver of the cellular decline we associate with aging and disease.
That sums it up perfectly. It shifts the focus from just energy output to the quality and accuracy of the manufacturing process within the mitochondria.
Which leaves us with a really provocative thought, doesn't it? If chemically forcing a complete shutdown of the mitochondrial engine using something like CA can actually reverse molecular aging signs and chronic inflammation linked to faulty mitochondrial translation, then maybe future therapeutic strategies shouldn't always focus on trying to boost or fix failing mitochondria. Perhaps in some cases, selectively silencing the most error-prone mitochondrial processes might be a more effective way to reduce the toxic burden and improve health.
It's certainly a counterintuitive but potentially powerful idea emerging from this kind of research.
Yeah.
A different way to tackle the problem.
Absolutely fascinating. Well, that's all the time we have for this deep dive. Don't forget to like and subscribe to support the SANS Science Experience. We'll see you next time for the next deep dive into the latest in science.